In the previous times, we've covered pretty much everything which was the first 15 years of informatics, okay, in two lectures. And this lecture will also go into 2 or 2.5 lectures, we'll see. And so the idea was, and around the time people didn't quite know what to make of bioinformatics, if you remember the Lincoln Stein message, which read, bioinformatics is dead. This is something I alluded to in the at the fag end of the last class of the class, the first lecture. And then, of course, that is because they said, okay, great, we've done all this development. Now what? And the answer to now what was, it just so happened, and that's why they said, well, there's really nowhere to go. And then the genome came about, and the genome was, people were working kind of in the siloed. One group was the one with Craig Venter, the Celera Genomics. They were using the shotgun sequencing, which is right now becoming, let's use the French term, de rigueur, as far as uh, the current next generation sequencing, and we'll look at that now. And the other group was, of course, the NIH-driven consortium group. And now what happens is they realize that you have one genome, and then there is so much going on because there's so much versatility in the process. There's so much diversity within genomes that looks like, and then there's of course the issue that there's something called as an ensemble view of something. And an ensemble view of something is, let's say, for example, when you go from this notion of this thermodynamics, right? They always like, well, what happens? What's the free energy change when two hydrogens get together? And that's really simplistic, right? And what is the free energy change? What is the enthalpy change? How does the entropy change? Then you have all of these notions which you may or may not have encountered at some point. The reason I use this word ensemble is because when you go from thermodynamics to what is called as statistical thermodynamics, then you're dealing with large systems, right? And so what happens with large systems is the same notions, the same entropy change delta S, the same enthalpy change delta H, the same notions of Gibbs free energy delta G come into being. They are also applicable, but the way they are calculated now cannot be restricted to these, one atom interacting with one atom, how much heat is released, how much heat is absorbed. You cannot work on that. So the thermodynamics then goes into what is called a large scale thing, and that's when you begin to use things like Boltzmann statistics, etc., in order to understand how these ensembles. So now what happens is, can you just say, I'm going to take a sequence of length 10 and then, blast it against another sequence? What kind of results am I going to get? Now what happens is you're working with something which is 3 billion base pairs, right? Just that little word that's been bandied about whenever we talk about the human genome. Oh, 3 billion base pairs. Okay, fine. So, and some of those are non-coding, some of them are proteins, some of them, obviously, now we begin to understand that there's a lot of complexity. Now what happens is you go from looking at something in the single sequence, just like we did in the previous two lectures. So now begin to look at something at a genomic level with all the diversity within it. Okay, and that's when you realize, oh, 25 years after they said that bioinformatics was dead, and used to use Lincolnstein's words, bioinformatics is alive and kicking. Okay, and I think this is what these two they will give you a flavor. There'll also be a teaser in there, in the sense that you are going to learn about uh, things like uh, other courses that are being taught that go in depth into things like next generation sequencing. I think Dr. Mohammed Fokar, teaches a class on transcriptomics in addition to advanced plant biotechnology. Dr. Sunita Sukumaran teaches a class in next generation sequencing. Just to take this further, Dr. Isabel Castro, these are all faculty at our center, teaches a class in animal genomics, and then another lecture on cell culture. So again, depending on where you are in your training, depending upon which way your PhD is going, you can take all of this. So, obviously you cannot cover all of next generation sequencing in 2 lectures, but I think, but I'll give you enough of a flavor that if you want to delve deeper, but at a bioinformatics level, we'll begin to understand a lot of this. Okay, so what is sequencing? This is everybody understands this at various levels. And when we go into sequencing, we look at sequencing, kind of take a step back and look at sequencing from from the early stages. Okay, so what is sequencing? Again, what is the point of the bioinformatics of genomes is to use computational methodologies to begin to understand things like sequences of genes, differential gene expression, and with a large component of it being used for clinical studies. Okay, so what is sequencing? Sequencing is a method to determine the exact order of nucleotides or amino acids in a DNA or protein sequence, respectively, right? What is the order? Now, the order itself is a major combinatorial challenge because the order of the sequence is there's, for example, there's only four ACTG, right? If it's RNA, T is replaced by uracil. You already know that from the previous lecture. But the question is, what is the order? Now, if every DNA sequence was four, you would say the arrangement is in the arrangement can be done in 4 factorial different ways, right? And which is 100 and what 4 times 3 is 12 times 2 is 24, right? So 24 different ways you could organize A, C, T, and G, but that's not the point. Is because the moment you go from 4 factorial to 5 factorial, when the sequence is 5, that means you have 120 choices, right? And or if it goes to 6, it's 720. But imagine if you had a sequence as long as 3 billion base pairs, right? Kind of it's an astronomical, beyond astronomy. This is now where you begin to say this can only be conceived by the power of thought. Okay, now, so what happens is if you have, so now what happens is it's a big combinatorial challenge, and then the fact that things are, things are at variance with one another, right? So now what happens is, so somebody says, here's a sequence, here's my DNA sequence, or extracted from something, or you extracted from something, and we'll have a small slide about this notion of sample preparation, and then you take that all it is a several microliters or several picoliters of a colorless fluid. From there, you've got to discern what is the sequence. That we know that it contains A, C's, T's, G's, but we've got to figure out in what order they are placed, right? And that was just kind of the reason I gave you this notion of factorial was because I wanted you to get a sense of the enormity of the problem and the challenge of the problem. Okay, so you have ACTG or you have 20 amino acids. At least we'll go with the conventional amino acids. How are they arranged? How are they organized in a protein? Again, it's a big challenge. So let's say, for example, you have this sequence, and it comes from a particular species. How do we know what the sequence looks like? How do we determine that really there is a T that proceeds from an A, and there really is a C there's really a G that precedes a C, and so on. All right. So the basic conception, again, I'm really sorry about this, but uh, the sound that you're hearing is something I cannot help. It is the they come with the leaf blowers outside, and it's always at this time, and so it'll end in about 10 minutes. But there's really nothing I can do. So do bear with me. And the worst part about all of the sound is that I have become so used to it. I've become so inured to it that the other day I was in a Zoom meeting with somebody else, and they're like, "Hey, there's some sound like a drone like sound. Can you please stop it?" Okay, so do bear with me, and I'm really sorry about this. Okay, so let's say you have a sequence, and the trick is one of the ways you do it is you break the sequence into smaller parts, and then you try to put them together. Okay, because it's easier to look at small parts. So think about it as a jigsaw puzzle, right? A jigsaw puzzle is a sum of its parts, and it helps that you have a picture so you know exactly how to assemble those pieces. And that basically is an analogy we'll come back to when we look at RNA-seq. next generation sequencing. Alternatively, then you have what is called as de novo sequencing, which is sequencing from scratch. And the analogy for that is, imagine you had a jigsaw puzzle and you had to put the pieces together, but you didn't know what the end picture was going to look like, right? So when people went into the creation of the genome, there was that's why, you had several publications of the human genome, and you had to have several publications because every time they published it they made corrections, right? So around 2000 is when the first genome, the human genome, came out, and after that they've had subsequent genomes, and I think now the 2022 is called the T2T. There's a telomere to telomere. They have supposedly done a comprehensive version, but that depends. And there are several schools of thought which will say that yes, but is that a true representation of the human species, right? And in order to address that, you now have the 1,000 genome, which actually is about 2,600, which is a very intelligently put together 2,600 groups of people from various parts of the world, which a combination of admixtures, a combination of people who are mostly purebred in the sense that they do not have a lot of they do not have a lot of outside breeding, and then you had communities, so very intelligently designed, and obviously even that is not a thorough picture. So I think somewhere in the UK, if they've not already begun this, they've started this notion of the 100,000 genome sequence. Okay, and I know because I've actually worked on one aspect of the 1,000 genome sequence with a collaborator, and I might be going to give a talk, international talk later this year again, but it's relate, and again I can tell you, because last time we talked about olfactory receptors being something that I'm interested in, it has something to do with olfactory receptors. Okay, so let's go back in time a little bit to the 70s, where you have, because we're looking at sequencing, in order to get a sense of how the process of sequencing scales up at a genomic level, let us look at the sequencing, and this is where you have a group called the Maxim Gilbert method, and then there is the Sanger sequencing method. Now, if you've not heard of the Maxim Gilbert method, you certainly have heard of the Sanger sequencing, because that was a pretty unique. And I think both this group, the Maxim and Gilbert and Sanger, got the Nobel Prize. So again, you can see the original paper here. So let's take a look at what it, and again, you don't have to remember this in great detail other than kind of some foundational ideas. So I'll just introduce you to the Maxim Gilbert. So the breaking up of the DNA sequence is achieved using 4 different reactions in different reaction vessels that give different fragment types. So let's see what that means. It means that you break it up, you break up the thing using organic chemistry, chemical methods, in very, very systematic ways, such that you know what the ends are, and then those ends can be juxtaposed to give you the full sequence. Okay, let's take a look at it. Okay, so how do you break it up? And so you have 4 different reaction vessels, and it kind of makes sense because you want to identify something that ends in an A, something that ends in a T, something that ends in a C, and something that ends in a G, or begins with. Okay, all right, so the first step is you block the 5' end by P32, which is radioactively labeled because the abundant isotope of phosphorus is P31. So you give it an extra neutron, and you now have an isotope which is radioactive. Why radioactive? Because it's unstable, inherently unstable. So you block the 5' end, the phosphorus, and remember the structure of DNA, right? We have the phosphodiester backbone. So in one reaction, you put formic acid to break or depurinate. So now what happens is, when you depurinate it, remember the purines, adenine and guanine. So you depurinate the 5' side. So you basically break apart the A and the G. Okay, the 5' side, which means that once that a top part of it is broken, you know that the starting nucleotide is either an A or a G. That's the first step. Again, it's a very systematic process. So now what happens is, because you have a group of A's and G's, you want to then re-identify this with how do you separate the guanines. So now, because remember the difference between is an excess methyl group. Again, go back to your pictures that we looked at in lecture 2. And then you want to now say, well, yes, but what if I just had a bunch of A's and G's in the same reaction vessel? Now you have some way to separate the guanines. So you have by dimethyl sulfate, you methylate the guanines. And then in a third reaction, you use what's called hydrazine. Don't worry about the hydrolysis. This is breaking of bonds with the pyrimidines, that cytosine and thymine are hydrolyzed. That means you do something. You kind of basically rupture the bond. And in the fourth reaction vessel, you add sodium chloride, and that prevents the thymine from reacting. So again, in a very systematic way, what you're going to find is that you're going to break these up in ways that you know for a fact that your first sequence, that your first nucleotide in the sequence is definitively something. Okay, and that's how you break it in systematic ways, and then you put it together like a jigsaw puzzle and see the answer you get. So here's the chemistry. So this is the depurination. Okay, so the purine group is separated from the ribose sugar group by using water. Let's not worry about the organic chemistry, and you separate it out. So what it means is that the purine group is separated from this group. Okay, so that means, we'll see what that means. In the second case, what you see is dimethyl sulfate. So you have adenine. You see adenine and guanine, and adenine and guanine, there is a nitrogen here. Okay, and there is a nitrogen group here, and then using dimethyl sulfate, you can replace the methyl group here. When you add in a very specific way, typically when you add a dimethyl sulfate, it'll add, in the case of adenine it adds here, but in the case guanine it adds in a very specific way. Then you use the hydrolysis using hydrazine, and you separate out. You separate out the purine group. See that you separate out the purine group here and here. You separate this part. So this is, you can see that this is where the cuts occur, right? So now what happens is once you knock that part out, further reaction is not possible because the DNA chain builds up this chain, right? The moment you knock it out, this gets stuck to the DNA, and then nothing else remains back. Okay. So now what happens is, you know that at the end of the first step, you're only going to get a bunch of A's, and I've not really done this nicely, but it cuts either at the A's, in front of the A's, and the front of the G's. Now, because that doesn't mean every one of these will cut. They'll cut in, like, in a kind of a random way. Okay, because if everything was going to be cut, you'd end up with these pieces that are so tiny that you can't do much with it, right? So you're going to get different places, but everything is going to be an A or a T. And then you can cut it on the C and the G, and you can see, for example, here the cut occurs on this side of the G, on the 3' end of the G, and you're going to get pieces like this. Okay, and so basically, again, I'm kind of hurrying through this because there are different ways in which you can cut these. Okay, and I'll quickly get through this, and then I'm skipping to Sanger sequencing now. Okay, so two things that to remember in Maxim Gilbert. Maxim Gilbert is a destructive methodology. Destructive doesn't mean bad, in the sense that your DNA molecule is going to be destroyed. Now the question is, why did I abruptly stop? It's just like it's systematically cutting at various parts, and you get a bunch of fragments. Now, the reason I stopped it and I'm going to Sanger sequencing is primarily because once you eventually put the sequences together to come up with your original sequence is exactly the same. Okay, so the take-home message from Maxim Gilbert is, you use 4 different reaction vessels. In one case, you know that you're going to get everything that starts with A and G. In the second vessel, you're only going to get things that start with a G. In a third vessel, you're going to get everything that starts with a C and a T. And in the fourth case, you're only going to get things that start with a T. Okay, so now what happens is, you have these pieces of varying length, because in every one of the reaction vessel you put the exact same sequence, right? So because they are being cut in these systematic ways, they can then be put together, and we'll see how. Now, the Sanger sequencing methodology is also called as the is also called the chain is also called the chain termination method. And the reason it's called the chain termination method is because it's a non-destructive purpose the process, and you are uh, basically building a complementary chain. Okay, you are building a complementary chain again in a very systematic way. And the complementary chain, then once you know what the complementary chain is, you say, okay, because I know what the complement is, I know what the original sequence is. Okay, and that is simply because the complementarity is sacrosanct, right? The complementarity between A and T and C and G does not get violated. In the case of RNA, you will see when we do the structures lecture, you'll see that there are some variants of amino acids, and then there are wobble pairs where there is a destruction where you kind of violate the complementarity for, but not with the original A's and the T's. These are just modified A, T's, and G's. Okay. All right. So the concept is different. The basic idea is still the same. In one case, you are chemically breaking apart the DNA in systematic ways. In the other way, you are in the Sanger method, you are building a complementary sequence in very systematic ways. Okay, so rather than break up the DNA sequence using chemical methods, formic acid, dimethyl sulfate, hydrazine, sodium chloride, etc., a DNA chain is selectively built by using the enzyme DNA polymerase, starting from a DNA primer. Let's see what that is, right? So let's look at some definitions, and you can look at this too. A primer is a short new. It's like priming. You prime the pump, right? Let's say you want to suction off water, but then you fill it with water so that by using the principles of the capillary action, you can get the pump to rather than pump water in a dry, just from a dry thing. So that's what's called priming. You're priming the process. You're beginning the creation of a DNA sequence by this primer, such that the DNA polymerase then comes in and has something to found itself on, to base itself on. Okay, so that is a primer, and again, you can go through the slides of the movies, and you'd be able to see this full sequence, just in case you didn't know. Similarly, just a little bit of DNA polymerase, and this all comes from this kind of it's a glossary from that Nature has the Nature journal has created. What is DNA polymerase? DNA polymerase, using a primer, it's an enzyme which basically facilitates the building of a DNA sequence complementarily, given an availability of nucleotides. Okay, all right. So then what happens is, while you're building this, once you prime the complementary chain and use DNA polymerase and a bunch of nucleotides with some special nucleotides, you're actually going to block the creation of an additional the propagation of the chain, again in very specific ways. And they are done using what is called as di-deoxynucleotides. So we have deoxynucleotides is where the OH group, the OH group from RNA on the 2' of the ribose sugar is replaced in DNA without, it's just replaced by a hydrogen atom, and the O group is taken away. The dideoxynucleotides have the oxygen group from the 3' group taken away, and the moment you have something like that inserted, it's going to shut down the chain propagation. Again, now some of you probably already know what Sanger sequencing in various things, but it bears repeating. Okay, so much like the Maxim Gilbert sequence, once you have fragments, you run them using gel electrophoresis, which separates based on mass, and then based on the mass and knowing exactly what the sequences are, you can then actually these are the pieces of the jigsaw puzzle that you can put together. Okay, so you can put the sequence back. So Sanger sequencing requires what is called a classic chain termination method. So you have your DNA sequence, then you have a DNA primer, and then you have DNA polymerase, and you have regular deoxynuclear triphosphates, and then modified, which are nucleoside triphosphates, and then one of them terminates the strand elongation. So here's a dideoxynucleotide, right? This is the deoxynucleotide, and so you can see there, okay, that is the 3' end, that's the 5', that's the 3' end, and what you will see in a deoxynucleotide is the difference between RNA and DNA is like the H group, OH group is taken away here, but here what happens is the 3', so 1, 2, 3, 4, and 5, right? So the 3' end, on the 3' end. So the moment, see, the 3' end is always required to form that ester, okay, and propagate the chain with the bases being on either side, on this side here, right? But the moment this O is gone, that means their chain cannot continue to propagate anymore. So you put a bunch of dideoxynucleotide. Now, even the dideoxynucleotide triphosphate, they are still driven by the complementarity. So a dideoxyadenosine triphosphate will still be complementarily interact with a thymine. Okay, so here's the sequence, the total step. You have 4 sequencing vessels, each containing the DNA template, the one that you've been given, right, whose sequence is to be determined, and there you put some DNA polymerase, you put a primer and the nucleotide. And once the primer starts the process, DNA polymerase will take the nucleotides and complementarily it will add to, to the original template. But then what you do is you also add, so instead of DATP, GGTP, you also add DD. This is the dideoxy. You add it to the vessel, but not more, not a lot. You have to add, the regular nucleotide triphosphates have to be put in great excess, right? Because what's going to happen is if you put these in excess, they're going to immediately go to the template and just completely shut everything down, right? Shut down the propagation. So you want propagation to occur in systematic ways, and once suddenly a deoxynucleotide shows up, and then just propagate the chain. So now what happens is now you have a shortened complementary strand. So you have one of different sizes, and then you can put them together like the jigsaw puzzle. The resultant fragments are denatured and subjected to gel electrophoresis. Now, there are some good videos to see it, which actually in a kind of a graphical way will explain this. It's a very interesting movie how that works. Either of these would be great. So I'm just going to leave that for you for your own, so again, whatever I have said, but there's no Maxim Gilbert. These are for the Sanger sequencing, and for the large part, Sanger sequencing became the go-to. But again, it's very, again, some of you have done any kind of undergraduate biology. Protein sequencing was determined in so many different ways until the Merrifield solid state methodology, solid substrate methodology came, and then it seems for the rest of us mere mortals, it seems like, wow, this seemed simple, right? And it's the something that profundity that comes from simplicity is what does not occur to everybody else. Okay, let's go watch the movie. So now what happens is, so you've got whether it's Sanger sequencing or the thing. Now what happens is because you've done these systematic cuttings in the 4 vessels, right? You're going to get these pieces. And then you're going to run them on a gel, and you know exactly what mass there is, right? Mass based on what it is, and you know exactly how something is going to start. Okay, and so you'll see that something that's of a larger mass gets held on the gel electrophoresis column a little more, whereas something that is of a lower mass will and of course you're denaturing everything, and denaturing is important because you don't want any folds, any kind of globular folds, and this is more evident when it comes to proteins, globular folds kind of getting stuck in, right? You want everything to be completely denatured, you want all the tertiary structure to go away, and then you can do a very specific system. So now what happens is you have all these pieces together. Once you have all these pieces together in very systematic cuts on this side, right? Then you can say, okay, this part may be belonging to this, and because you know exactly what sizes these are. right? Eventually, it reached a point where, like Sanger sequencing, the process was automated, and you just, so you can use, and actually, as our center, we used to provide services, but then everybody else was providing the sequences for cheap, so we, okay, so that is again, like I said, if you're going to talk about sequencing, let's take a step back and begin to think in terms of, like, what are the origins of the sequencing process. Okay, so again, three of these guys won the Nobel Prize, certainly worthy of it, because, again, in retrospect, but a kind of thought process that goes to intellectualize and conceptualize something like this is not, it doesn't occur to everybody. All right, so now we can go and take a look at the UCSC, the genome browser. Okay, now I'm not going to delve too deep into the genome browser, in the sense that we can go take a look at it, and then, there are many different ways in which you can do different things. The genome browser evolved into many things, and so we can just I'm just going to copy the genome browser, and we can go take a look at it. But then again, depending on what you want to do, there are many videos and things like that that help. Okay, so this is the genome browser, and this is again a very useful utility developed at the University of California at Santa Cruz. So it's called the UCSC genome browser, and you'll realize that when it comes time to things like searching, right, and rapidly, and that's basically very genome focused, you will see that this can be used. You can do primer pairing. Again, this is one of those resources that every time you go, you're going to get new stuff in there, right? And it has many others. So, but this was their primary thing, and of course, continued funding will allow you to kind of learn more and more about it. So once again, I don't know much about this, in the sense that I do not use it for my personal research. If you do, you're likely, but at least let's give ourselves a bit of a start. Now they have all the typical models, so we'll just take a look at some of this. So you have the human, mouse, rat, zebrafish, fruit fly, Drosophila. Then you've got C. elegans, and then you've got the yeast. So the typical models are here. You can actually go up, and then you can see that they've got species connected, and there are all kinds of assembly hubs, different kinds of species within each of these categories. And so you can see that, and every time you could potentially look at a sequence, and each of these genomes, there is comparison, right? So there are different assemblies, if you can, depending upon. So this is the 2013. Now it's 12 years too late, is the one that is the most stable, let's call it stable. But you can see that if you really want to do a comparison, you have from the build number, it's called build. Oh, you can't see it, but if you were on that website, and I would certainly encourage you to do it. If you like, I can drop down and I can say, okay, I want what version, and you'll see that change, right? So that's the 2003 version, so that's one of the earlier versions. That's the build. Okay, and then from 19, which was build number 37, that you can see here, you can go to the one that was really stable, okay, and which is considered the default. And then here you've got the T to T, the telomere to telomere assembly of the genome, right? And now you can do any search. You can do a search from a chromosomal level to a chromosomal level. So let's just take the default. Okay, let's take the default, and I just added that, and you say go, and you see that here is that part of the genome. It'll tell you that this is the Q branch of the chromosome. It tells you that this is the P. I'm sorry, this is the P branch of the chromosome. This is, I'm pointing out, is the Q branch of the chromosome. And then you can see 3 prime, the little arrows here. So you can see the arrows point whether it goes in the forward direction or the reverse direction. Right, and then you can see all kinds of interesting things. This is the the GenBank entry for this. If you click on this, you're likely going to go directly to GenBank, or it stays some indication of the browser. You can do things like you can do things like highlight. You can let me go up here. So you can see here, you can start a tutorial. You can change this range. It tells you this has what you see here is 36,747 base pairs long. You can actually put your own your own gene in here, and it'll tell you what that gene is. Um, Again, so in this chromosome, what are we looking at? You see this little red line? That is the part of the chromosome we are looking at. So you could see saying, can you jump to a new position? For the time being, I will not. But these are the parts of the chromosome, right? You can kind of see, I can highlight a part of the chromosomes, like here to here, for example, and it says, do you want to jump there? Right? I can, so it basically you can jump to different parts of the chromosome. Now here what happens is I clicked on something and it gives me all the SNPs. You can see all the SNPs that are there here. I'm sorry, here are the SNPs that I'm pointing at. And each of these SNPs has, you can see the GenBank entry. So if we click on the GenBank entry, it doesn't go to GenBank necessarily, but it'll go to more information. So again, a lot of the times you'll see that a lot of the web development that takes place has at its core uh, this notion of interoperability, all this this notion of knowledge management. And knowledge management is actually very crucial because do you need to reinvent the wheel? So for example, if I say I have this sequence and I want to find out what that protein is, and so does it have a structure? Does that structure go to the protein data bank? Or does it have a structure that goes somewhere else, or does it have a structure or it has a sequence? Can I look at it in the genome browser? Can I look at it in the ensemble genome browser? All of that information, all of that information can be seen here. So this tells you more about that particular chromosomal. Okay, so we're obviously looking at chromosome 9. That's the GenBank ID. You'll find that information in here. It tells you that, hey, there is a translated protein from the genomic DNA. There's a predicted rMNA, right, mRNA. There's a genomic sequence that you have. So all of this additional information will be provided for you kind of in an automated way. So if we go back one, I can kill this. And now we're back to the original file. So again, by people who run this, lots of tutorials on how you can use this. One of the things that is really useful here is these things called tracks. Okay, so here you see a track, and I can right click on it and I can say hide this track. I don't want to know anything about this information. Hide it. Okay, now you can do something like add track. So if I look at genome wide association, which is currently hidden, right? I can click and say make it full, and then I go here and I say refresh, right? Refresh, and then it'll give me some genome-wide association studies. It'll give me a whole bunch of things. Now this is just so you can see a whole bunch of things based on genome-wide association studies. All these so-called SNPs, these changes, what kind of changes occur across various. So this tells me it's a T to C change, right? That you can see somewhere else is an A to C change, an A to G change, and all these things. So these are the SNPs. Where do these SNPs occur? Again, the idea of genome-wide association study was to look at that there's an association of genes or SNPs, or basically genetic structural variants which actually change. So all of this information. So this is a lot of information. The idea is not to necessarily cover it, but again, there are a lot of YouTube videos. So if you want, again, this is to point this resource out to you, that if you wanted to do something with this in more detail that would immediately help your research goals to find out something more, then this would be the place. This would be the place, and this is where you would come to. Okay. So if you look at the PowerPoint, I have given you here some links to tutorials. Okay. Alternatively, if you go here, you will see that again, these are all tracks. You can add tracks, reverse the tracks, add custom tracks. More and more tracks, more and more information can be made available to you. So now I see this genome-wide association track, which is here, and I can go to hide, and then, so I don't want it anymore. See, genome-wide, it was full, went to hide, and then I go and click refresh, and you'll see that now it's gone, right? It's gone. So again, here there's a tutorial which tells you how to do this, how to run this thing. There are lots of videos which tell you what to do, how to do it. And again, to me, from a perspective of this, if you want to start looking at things at the genomic level, this is a great place to start. All right, let me look and see at the teams whether, okay, so nobody has sent a chat, so okay, fine. Good. Sometimes there are so many windows that I'm never really sure what I'm looking at. So, okay, the PowerPoint. And you'll see here that in the PowerPoint, here's a bunch of here's a bunch of videos that will get you started. Now this one, see, it's actually a channel, not just a specific video. It's a channel, so it's got a bunch of videos. They keep adding to it. Sometimes just a search within YouTube or a Google search, if you want a particular thing in the UCSC Genome browser, then you click on it and it'll be able to take you there. Now here I will not go into this genome browser because again, you'll see that you can get to the Ensemble genome browser. Okay, that is the European Bioinformatics Institute. They have a genome browser of their own, and they are you'll see a lot of information. It's not as popular. It might not seem as intuitive, but they have a browser of their own, and here you can see a bunch of YouTube videos, tutorials, etc. As to how to use the Ensemble genome browser. There is, however, there is also a genome browser that is in GenBank. So if you go to GenBank and click, let's see, I'm just going to check on this to see if it works. GenBank overview, NCBI GenBank, and if I click on like a gene, so let's see, factory receptor search, and I don't know what it's going to give me. Okay, so let's say it gave me this is the entire chromosome sequence, okay, of from Bos Taurus, which is cow, and if I click on gene, okay, so this is the full annotation, etc. But if I click on something called a gene, so you can see the assembly here. The assembly here, you can view the annotated genes. Oops. And so all these are IDs. So for example, you can take a look at it. You can download it. You can also, I hope, yeah, you can download this information from the cow. Okay, this particular chromosome, this particular build, and you can look at various things here. But I think if you look at a specific gene, when you go to GenBank and look nucleotide, you get a lot of information at a genomic level. An entire chromosome, for example, or a big cosmid. Uh, so all of this is chromosomal information. But if you click on gene, you actually can get the GenBank um, genome browser. However, it's not as popular. For example, a lot of effort has been put into, and there are some other resources like something called the structural genome browser, where it's kind of the view that you get is three-dimensional nature and things like that. So there's a lot of interesting things that have been done here. But again, this is just gets you started. Some of you might never need to use it. Some of you might. So this is a good place to start. All right, let's begin to think a little bit about now. These are not as popular as they were before, but when we start thinking in terms of what are microarrays, okay? So microarrays were all the rage somewhere between 2003 and we started around 2003. Why? Because this was the first time that you could do information, you can get information, especially with a lot of clinical significance. You could do this at a level of a tissue, level of an organ. So rather than do one sequence at a time, one gene at a time, look at the expression levels of something, you could do an expression level in a at kind of in a ensemble way, at a you know several thousand at the exact same time. Okay, so microarrays became really popular, and there was a company. Uh, and I'll tell you what that name of that company is. They were like the industry leader, like the Illumina and the PacBio of our times, um, and then they kind of fell by the wayside because the methodology was supplanted by next generation sequencing. So microarrays, okay? So microarrays were the first example of what is called high throughput studies. It relied on hybridization, partial hybridization, complete hybridization. So what is hybridization? When you bring through DNA sequences that are complementary, they automatically bind again because that's the nature of complementarity, and that's kind of the very origins of the very specific, the singularities of life, to speak, and they resulted in a complementary. So that is called hybridization. So now what happens is if you have some sequences that are not completely, that an A and a T will always bind together, but if it's partially hybridized, completely hybridized, if the sequences are completely different, so you get nothing, then you get no hybridization. So when two complementary sequences are brought together and then they form the double-stranded DNA sequence, it is a called hybridization. So you could literally do several hundred thousand simultaneous experiments, right? If you wanted to expression levels or binding, because you could do it in different ways, and it can be used in several different areas of research, with the clinical being very important, and we'll see what we're talking about there. And it gave rise to biostatistics. So before, what used to happen was, now statistics are really very important because we'll see why, but I'll just give you a little bit of a teaser. Why were they useful in biostatistics? So biostatistics is a uh, before, what used to happen was, yes, everybody needed statistics, right? At a foundational level, some of you have said, well, we did some work, we did some bar graphs, and we did a standard deviation because we needed to know, the plus minus, what are the probabilistic range under which those results could occur, right? I mean, everybody, most of us, at least I'd like to believe that everybody knows that you know exactly what the formula is in Excel. If you have a number of terms, how do you calculate the standard deviation? That's another way to determine. Now, when you when you do something ensemble, right? I gave you the example earlier that if you had, wanted to do thermodynamics and determine pressure, temperature, or use those, right, in a, like when you wrote your chemical equation on a piece of paper, versus now imagine that in a real life situation you have 100,000 or 25 million molecules working together. Now you want to calculate the temperature or the pressure or the heat energy or the entropy change of an entire system. That's when, just the little thing that you wrote works well for two molecules. It doesn't work well for 25 million, and therefore you had Boltzmann statistics, etc., which were used to determine some of the instance of information. Now, what happened when you had microarrays was, it was a new technology. Everybody who could afford to, get microarray work done was able to spend a bunch of money, get the work, get the results, publish the results, talked about this expression and that expression, and these proteins are overexpressed and these genes are overexpressed, especially in cancer, for example, versus. And then what they found out was, one of the things about Okay, I've already said this. If somebody says trust the science, they are not a scientist. They are a charlatan. Okay, that's a political statement. Fine. What you do is you trust a scientific method, right? You do something. You have a hypothesis. The experiment has to be tested, right? After it has been tested, you're going to get results. In a completely different environment, those results, that experiment under the same experimental condition, it has to be replicated, right? Under the same conditions. Or if the results are not the same, then at least you have to articulate why the results are not the same. And one of the reasons are that the conditions under which that experiment was done were not exactly the same, right? The idea is replicability, the idea is reproducibility. Okay, so what happened with microarrays was that it was large amounts of data, large amount of data. Experiments were very popular, and what they realized was that the data was not reproducible. Different conditions, different, everything was spurious, and the reason was because in an ensemble calculation, you have to be driven by statistics. As I said, there's thermodynamics and there's statistical thermodynamics, right? In which case you have to be able to not reproduce a result, but every result has to be has to have associated with it this notion of probability. There is a possibility of an error. Okay, you cannot walk away without but saying that this is error free, that even if you say that you're certain about the results, there's no what's called metaphysical certitude. You are driven by probabilistic, right? Nothing is deterministic, especially in an ensemble situation. Your errors are, that's exactly one of the reasons when we looked at the example of a log odd ratio. And I didn't even know this when I was one of the earliest people to publish a heuristics determined AI paper in neuroscience, but in a very narrow focused way, so to speak. It wasn't a program that anybody could use everywhere. I mean, it did quite well, but then they said, okay, in this particular paper you have in this particular paper you have to extract from the neuroscience literature publications that would be useful to be put in the database. Okay, so the simplest thing is you would say, well, I'm going to get something called as false positives, right, where I say that something is should be put in that database. So what we're trying to do is we had a database, it had a certain architecture, and we was trying to say that I want to write a computer program that will actually make a determination by looking at the abstracts of all these papers, let's say from the Journal of Neuroscience, and it is worthy of being deposited in this database. Now, the worthiness was determined by experts who had a combined 70 years of experience in neuroscience, and they kind of determined what needed to go in there. So at the end of it, when I did this work, I said, okay, now you have to tell me whether I'm right or I'm wrong. So now what happens is you say, if I have a false positive, which means I said, hey, this paper needs to go in there, and they say, nope, it's not, right? For whatever reason. So let's not worry about the reasons because they went beyond the objective in the sense that they use a lot of experience. Oh, I don't think this needs to go in the paper because 30 years ago I know something about this. Now, the computer program that I wrote had to work on the paper it saw. It did not have the advantage of 30 years of experience. So that came from somebody's knowledge which was stored in their head. It was not on the paper. Okay, so you expect that. Now, let's put that in the context of and then what happened was there might be something which my program decided was not supposed to be in the paper, but the gold standard, these experts looked at it and say, hey, you missed a bunch. That's a false negative, right? So everything has a statistical notion. Now, let's take this work that I did and put it in the context of the log odd ratio. Now, when we looked at log odd ratio, what did we see? We saw log odd ratio, or just let's take the log out and just keep it as the odd ratio of and we use the example of a disease. And the exact same example, the odd ratio, was used in the PAM matrix as well as the Blossom matrix, right? In the sense that if you took a family of genes of certain diversity, you say, what was the probability of an adenine being, or an alanine being replaced by a glycine, right? So you could say an alanine was supposed to be there and it was replaced by an alanine. That is one, right? The other one was an alanine was supposed to be there and it was replaced by a glycine. Then you have an alanine was not supposed to be there, but it was replaced in that position by an alanine. And the last case was an alanine was not supposed to be there, but it was not replaced by an alanine. Okay, so now what happens is when you look at all of these comparisons, you got your entire Blossom matrix, which tells you probabilistically what is the chance of that being in the right place. And those are the numbers you used in your BLAST search, right? Especially in your protein BLAST search, BLAST P. We looked at that last time. Also, now take the odd ratio notion before I come to what I did in the paper. And I didn't know anything about odd ratio until somebody said, no, 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 if you really want to know how good your program, how well your program is working, is you need to have to calculate an odd ratio. So I had to go look it up and study it and then, do the calculations and things like that. Now, when you look at odd ratio, what else did we do? We look at the disease, right? The disease was what? Okay, let's take whatever disease and a particular symptom. So it's like you have the disease, you've been diagnosed with the disease, and you have the symptom. That's one. You were diagnosed with the disease, but you don't have the symptom. That's the other part of that block of four in the matrix. Then you were not diagnosed, but you have that particular symptom, and then you were not diagnosed and you don't have the symptom, right? That's A, D, C, and G, B, and C, and you take the ratio of that. That gives you the log odd ratio. Now, extracting it, and again, I'm trying to make a long story excruciatingly long, but again, I'm trying to tell you that I say I wrote a program, it can find journal articles, and those journal articles can be deposited in this database. Well, and that's why we have to stop and we say how good my program is based on probabilities. Okay, so what happens is, now I found an article, it's supposed to be in the database. That's one. I found an article, but it's not supposed to be in the database. That's two. Well, I did not find an article that was supposed to be in a database. That is three. And then I did not find an article and it was not supposed to be in the database is four. Right? So now what happens is, now we are beginning to take, and when you add the log odd ratio, that the example that we used from Wikipedia was about what happened to men versus women. How much do they drunk? It's 36.5, but when you took the log odd ratio is 6.5. It's a lot more manageable number. Okay. Now, so it gave rise to biostatistics. Now the biostatistics might tell you, I see hybridization, right? And we'll see what that is. Now we'll actually look at some graphs in the upcoming part of the thing. I see the hybridization, and the hybridization tells me that it interacts with a cancer cell. Ooh, that means there's cancer, or the expression is overexpressed, right? Alternatively, it's overexpressed, but it's not related to cancer. Then it's related to cancer, but that particular gene is not showing me an overexpression, or it's not related to cancer, and I don't see that particular expression. You see, the whole thing is tied in. But the moment is when you just present this and you say, hey, look, here's the result, definite signs of cancer, or definitely no cancer, and then somebody else takes this data and says, hey, wait a minute, I see something completely different. Now what happens is you say, one of these are wrong, right? And therefore we go back to this, what is sacrosanct is a scientific method, right? And if you remember the book, the paper I gave you, I hope some of you have started reading it. It's the book by Henry Hazlitt, right? Thinking as a Science. Okay, so when somebody from one of the weather people come and say global warming is going to be the end, science is settled, that's it, you've got to ask yourself, probabilistically, right? For example, when this is more a political statement, when in 2000, when this global warming was the furor, everybody was running around, um, they said by 2015 there will be 10 feet of water in downtown Manhattan. Well, it's 2025, right? And so then you begin to ask. And for them it's like, well, the scientists have determined it to be so. Well, the science, or is it the scientific method? Is it reproducibility? Do you have data? And I really have to struggle with this in my own work because my work is theoretical. I'm a theoretician. I'm a bioinformatics person. When I write my papers, it's like, okay, all right, um, it's the best I can do in the sense that the methodologies I've used are state of the art, the computational facilities I've used are the state of the art, the complexity that I put into my thinking, and when I say me, I mean my students also, has been the state of the art. This is the most complex we can make the system, but can we really replicate a human cell? If we really replicated with everything we had, yes, we have done more than anybody else to further this notion, but can I just say, oh, that's how it behaves? No. So in my paper, I always have to put all these caveats. I have to say, this is what we have. These are the conditions under which this work. If, on the other hand, somebody tries the experiment and says, well, we don't quite get the results, I will have to take a step back and redo my work or add complexity or say, I tried to simulate this interaction for 1 nanosecond. By the way, 1 nanosecond takes about 15 days on a supercomputer. No, that's not quite going to work. I might have to take 2 months, or I might have to go back to my code and say, is it properly optimized? Right. But when I say, nope, that's it, my computer says so, so the science is settled, that is a problem. Okay. Again, as you're budding scientist, as you go through your process, keep that at the back of your mind. And what happened was, just to illustrate a point, that it gave rise to biostatistics because now they said, nope, every time you have something like this, you have to do a statistical assessment, provide the results, provide the error bars, provide all of that. Now, what happened was that because it was a new technology, all these publications, people jumped on the bandwagon. They got published in all the top journals. Some of the papers had to be retracted. So the whole thing was a mess because you have to understand the foundations of it. It gave rise to an acronym called Miami. Full form MIAME, not Miami I like the city, but MIAME. And the full form of this acronym is Minimum Information, MIA, about A, the little a is microarray experiment. So minimum information about a microarray experiment, and that is what is called Miami, which means that they said, okay, great, you've done a microarray experiment, you have to meet certain criteria. If you don't meet certain criteria, we're not going to allow you to publish this paper. Okay, and you'll see that especially in structural organic chemistry, structural biochemistry, the protein data bank, etc., they're very, very, very careful about it. Okay, and you'll see what I mean by very careful and how they are very careful when we come to that. Okay, it's all probabilistic. Okay, so now let's take a look at the kind of the foundations of how a microarray experiment is carried out. So on a substrate, I'll tell you what that company's name is. On a substrate, that substrate could be a glass plate or polystyrene beads or this company, it starts in A, they came up with something. They came up with something a little more, a little more elaborate, but you had to have money. What you did was you took a bunch of oligonucleotides from a specific family, say olfactory receptor, and you attached them on that substrate. Now, this company came up with a methodology that allowed you to kind of do it in a better way. These are what are called as probes. Okay, so your oligo probe. Now, what used to happen was when you gave this company enough money, you could tell them how to design your probes, and then they would design it and give it to you. Okay, so they would, and those are called as probe sets. A probe set is whether it's a microscopic chip, like an IC, like an integrated circuit that they use in computers and electronics and everything. Or you could, and some people who couldn't afford it said, hey, you know what? I can do something similar, but I can do it on a glass slide, for example. Okay, or they could use like things like lithography, which is something where you use to embed something. There's all kinds of methodologies that were used, and I'm sure so this company, and the name will come to me soon. Oh yeah, it's Affymetrix. Okay, they were all the rage, kind of like Illumina is all the rage. So these are what are called probe sets. So these are oligonucleotides, which are pieces, right? Manageable pieces of various, for example, the olfactory receptor genome, the family came out, and then people said, okay, go to Affymetrix and order us a probe set of olfactory receptors. So they got a probe set of olfactory receptors, and they were able to do a bunch of experiments on it. Okay, all right. So now let's say you have a sample mRNA from a cell, and then you got that mRNA from a cell, and what you did was you used reverse transcriptase to convert it into cDNA, complementary DNA, and then what you do is you take that DNA and you wash it over. So you can do this for a cell, you can do it for an mRNA extracted from an entire tissue. If the organism is small enough, you can do it for an entire organism, you can do it for an entire organ. The entire gene profile, that is expressed gene profile, can be extracted, okay? And what you do is then you wash it over the probe set. And each of the cDNA is also labeled with a fluorescent dye, and the dye says that as you wash it over the probe, if that particular thing has a complementarity, hybridization occurs. If no hybridization occurs, it washes away. On the other hand, these are the pieces of grass, like pretend like they're pieces of grass. They're just floating around, and as you wash away an entire thing, they'll grab the gene sequences or the DNA sequences which are complementary with the probe, and they get attached to it. Now, because these cDNA are fluorescently labeled, you're going to see a spark of a light. That means hybridization has occurred. Now, if the hybridization is really solid, it's going to give you a different color. If the hybridization is partial, it might give you nothing. Sometimes, the probe might be bad, so it's not picking up anything. Sometimes you're not going to get it. But remember, if you're going to basically get the genetic or the genomic profile or the genetic profile of an entire organ, then you're doing thousands of these, right? And if you do thousands of these, you are going to get errors, right? Again, that is the notion of the probabilistic determination. Now, once you begin to understand it and says that yes, we are driven by the probabilistic nature of all of these things, then you can, honestly describe your results and walk away knowing that you've done the best you can. Okay? So you will see that double-stranded DNA formation, and these oligos are single nucleotides, all right? And then your DNA is single nucleotide, your cDNA, and you wash it, and then you wash the entire process. Anything that's not attached through hybridization washes away, and then the fluorescence is captured in an image. Okay, so you see these are the probes, they're a fixed set of oligos, and then you see the DNA that attaches, and you will see that here there is all these are single-stranded, and this guy shows up, and it forms a full complementary, and then you will see that it lights up in one place and in the next place. And now what the probe sets will tell you is exactly where it is attached. Okay, so you will see once again, you make cDNA that's complementary. Now, what you could also potentially do is you do the exact same probe set, and I'll now put this in the context of, let's say, for example, this is in the clinical, so let's say there is somebody who has a control, and the control will be somebody who is not, and you know exactly which oligos or little pieces of which genes are already in there, because that's what Affymetrix did. It actually designed the technology for you. Then once you have these are your typical genes, right, from a system, and then you get somebody with a control. That means somebody who is not diagnosed with cancer of that particular tissue, and then you have somebody who is diagnosed with cancer with that particular tissue, and then what you do is the probe sets are exactly the same, right? And now what you see is that you're going to get, depending upon which gene is overexpressed, you're going to get lots of it. And the one that you're going to get lots of, you're going to get probabilistically, you're going to get a better chance of hybridization. So in knowing that particular position, you're going to get a brighter light, right? And then what you can do is you can take the control, you can take the one that is disease, for example, and you can superimpose it. And in this superimposition, right? For example, which gene is upregulated, which gene is downregulated, and there are different ways in which you can look at it, right? And based on this, based on the hybridization, and then you can see that anything that's a dark area, which means that the level of expression is the same, right? So the colors cancel each other. And then on the other hand, you have seen, the green is downregulated or red is upregulated. How red, right? That is the question. How red? If it's very red, that means it's strongly upregulated. Is it dark green? Is it light green? So now what happens is, because you know what the probe sets are, they come from which gene, you get a picture of that upregulation of a gene. So it's a brilliant idea, currently supplanted by next generation sequencing, which can do it at a higher level, potentially faster, and we'll come to that, right? So now what happens is you have microarrays, and microarrays are a great way of looking at something like this, right? And what tells you, because the probe sets, in that probe set, you know exactly what is on what position. When you say exactly, I'm assuming probabilistically, because they have actually designed your oligos, so you know which genes they come from. Okay, so now what happens is you can go back here and you say these are, or some genes, they are highly expressed, but they're highly expressed no matter what you do, right? In both these, if they're highly expressed, which means that once you overlay these two slides, so to speak, or so two pictures, so to speak, they cancel each other out. So just because something is highly expressed, it could be what is called a housekeeping gene. It had nothing to do with the disease, right? And that's the notion of differential gene expression. That is what RNA-seq does, and we'll look at that too. So microarray analysis, a small number of experiments results in a large number of genes with varying expression levels. So again, it's a fantastically brilliant idea to say that I can take the entire breast tissue of an individual with cancer, or I can take the entire tumor, extract the DNA from it, extract the mRNA from it because it's already expressed. So as mRNA, it's gone through the transcription process, converted back to cDNA, and now I see the breast tissue. On the other hand, if I take a similar tissue from a control, somebody who's not diagnosed with a disease, I can see exactly what it is. And high expression doesn't mean much. Differential expression is what it's important, right? So some genes are housekeeping genes. So they are both highly expressed, but they cancel each other out. When one is high, one is low, one stays nothing and the other lights up, or the other one lights up green, that is where you can do it. Here's one who can determine the easiest context, and the reason it became very popular was because of this. But again, because this is a very small thumbnail of the entire picture you get, right? And secondly, here you can see, is it red? Is it how red? Is it really red? Is it kind of pink? Is it dark red? Is it light red? Now what happens is, is it like completely hybridized? Is it partially hybridized? Did 30 of these nucleotides, just one after the other, hybridize really nicely? Did some of it hybridize? And that is the crux of this work. Okay. So one wants the smallest number of genes. Obviously, you want a small number of genes because you don't want so many genes. You says, well, how am I going to manage all these genes? So you want a small. Why? Because you want biomarkers, right? You want to say those genes. We need to do something about those two genes, and then you might be able to basically take this in a mouse, maybe do a knockout. Where you take that gene out or make it non-functional, and then see whether that's immediately responsible for the breast. And then obviously in mice, because you can go through several generations, that thing propagates really nicely. Okay. So now let's take a break and then come back and look at David. Okay. I'll be back in a minute. This hard candy takes a lot longer to eat. Okay, so there is this resource called, okay, there is this resource called David. There's a classic example of it's a classic example of an acronym. Remember, I told you the difference between an acronym and an abbreviation. So the full form is Database for Annotation, Visualization, and Integrated Discovery. And it's a little annoying, but let's see. So let's see. Let me type it everywhere because, oops, um, oops. So David is David.ncifc. Okay. So the lull is because I'm actually trying to find out the link. Um, So you can see the warning is giving me connection is not private. Attackers might be trying to steal your information from step one. Oh, jeez. I'm gonna test something. Okay, so it's not my resource. I think they have a problem. Okay, so okay, so the best I can do is point you to two videos, and again, if you have any questions, let me know. So I'm just going to go to the web. Okay, I don't know what this is, but let's go lower, and I type this in. Okay, and you'll see that just type NCI FCRF, and it'll tell you how to use David for the functional annotation of genes. Okay, so just, and then if there is a video that I have put together um, where I explain this, it's a little better. See, so typically you'll see here, this is what most of the thing looks like. So it's almost like you put a gene and it's going to, so so again, this YouTube will give you more information than I ever could. And with as I told you about in terms of the interactions, I don't know why this is I tried a few other databases that you couldn't see and everything was fine, so I'm not quite sure why they only have a problem here. Okay, so the idea was the idea for David was we have a bunch of genes, lots of genes, right? And the only way you can get lots of genes associated with a particular condition was in a, at that time, it was in a database of um, genes, and that came from a microarray experiment. And so the problem with the microarray experiment, you got lots of genes. So you just cut and paste a bunch of genes, and what it would do is it would take those genes and give you one page where it identified with all the PubMed articles related to the genes. Another time it would go take a gene, and at the same time you could click on another link and it would tell you all the clinical work done on the gene. It would tell you things like all the pathway analysis that was done, the metabolic, the biological pathway analysis that was done associated with those genes. So if you have a big list, you clicked on one gene, it told you about the papers, it told you about the clinical trials, told you about the pathways, it told you about about disease associated with that. So basically, what happened was that David was one way in which you could take one instance of information, and then do it. Uh, Take one instance of information and then just kind of expand it into many bits of information. And the idea was to be able to do it in multiple genes at the same time In fact, you could actually, I'll show you this, the web here. Uh, I'm just going to show you the
Hit
first
it,
page.
Papa.
Ella tiene ritmo.
I don't know what this is, but fine. So you could see that there were all these genes, gene IDs. You could literally take, let's say, a probe set, okay, and the probe set was associated with a whole bunch of genes, and you could take that entire probe set and dump it into the annotation. So, for example, you see, and the reason, let me go back a little bit, and you see that I'm going to pause the video. You see that 80 was a probe set related to Affymetrix. So sometimes you wouldn't even have to have a list of genes that you would put in there. You just see, and see it says Affymetrix. It says Affymetrix here. It's not immediately visible, but it says Affymetrix probe set. So you clicked on the probe set, it knew exactly what genes were represented, the oligos were associated with what genes, and basically you could get the entire ensemble information based on that particular probe set. So I'm sorry, it's creating an issue. This has been a problem in the past. Once in a while David will act up, will not allow you access, and then I'll actually maybe even try it from home when I get back home, and we can and I can try it that way. So this is the uh, David, and gives you lots of information. I mean, you could sit there pretty much with one gene for hours. Okay, so it was designed to provide the information exactly in the way it was supposed to. So once again, I apologize. I showed you exactly what the security issue was, and we can't look at David. Hopefully we can look at the next link. Okay, so anyway, that gives you more information. But now let's say, for example, you have additional analysis, right? So now you've got the log ratio. See, 6 genes over 6 experiment. Here are the experiments. This is your matrix. Here are your genes, and these are your values, and those values are the log ratios, right? See, and the log ratio, again, as I told you, in 3 different ways. It is, you expect an expression pattern for that particular gene, you get it. You express a pattern, but you don't get it. You get something, you don't express a pattern, but you get that result, and then you don't express and you don't get that. You multiply that, you take the log to the base 10 of it, and you get this expression. So you'll see that 6 genes, right? Now, in a real experiment, you could have a probe set of 50,000 genes, okay? And that's one of the reasons why David was able to get all that information, and then you could clump all that information and look for similar instances of information, like, oh yeah, I did all these particular experiments and all these particular genes that were represented on my probe set. Here I go and I get all the disease for each of these genes, which of the disease are common, and then you would start beginning to build kind of a knowledge base of that information using it. So you'll see these experiments, right? So for example, you could have a healthy versus control, etc., and you see the expression patterns, and you do the log odd ratio of the expression patterns. Why? Because we understand statistically, just because the color showed up doesn't mean much. Now you'll see that negative numbers here, positive numbers here, and here you'll see there's nothing, which means that probe set was kind of at fault in that particular location. Now, if you look at this and you look at now, each of these genes is measuring the hybridization, right? And what these statistical samples are doing is they're telling you of the worthiness, the probabilistic worthiness of that particular color at that particular spot, right? Which is measuring the genes the against whatever experiment you're trying to do. So you will see here that this is then a graphical representation of this. And as you can see here, something was at fault. The probe set was messed up. The oligo was not showing up. Maybe it fell off. Maybe it wasn't attached properly. And you can see there's a gray area here, right? So if you see that now the log odd ratio, that means that the control and the disease or whatever is being tested are the same. are the same in the sense they cancel each other out. As I said, you might have a highly expressed protein, but in control, if they're both expressed really highly, that's a housekeeping protein that is not immediately associated with the disease. What you're looking for is a differentiation. And the genes that have the biggest differentiation, like, oh, it was nothing, it was like a very small expression pattern in a healthy person, but look at the expression level, they're off the roof in a diseased person. In which case you would say, okay, this is crucial, this is important, right? This is of significance, um, right? And so for example, green indicates a negative log ratio, okay? Red indicates a positive log ratio, and then you can see all kinds of ranges of things that happen. Light green means not as high, and this is a log 2. Now again, I said log to the base 10, it's log to the base 2. Why 2? Now this number 2, which is a log to the base 2, will rear its head when it comes to differential gene expression. What it means is, let's say something has a value of 10, okay? The other one has to be either to be considered upregulated. In the diseased case, that expression level has to be 20. Okay, now 10 and 20 are just meaningless terms, they're just used for illustrative purposes. So 10, 20 or above means log to the base 10 will say yes, log to the base 10 of 2 will say, log to the base 10 of 2, log to the base 2 of this particular ratio, if it's more than 2, which means, oh yes, that is doubly, it's a twofold change, which means an expression pattern at that particular gene for that particular diagnostic tool or whatever is now double. Okay, is double or more than double. If it is, or it is halved and less than half. And that is what's called the log to the base 2, which means that something is double. And if something is double its expression, okay, it is upregulated. And if something is half, it's downregulated. And that's a pretty good criteria. Now, if on the other hand you have many genes which meet the double criteria and you're like, wait a minute, I need to find something that's of real significance, you would say, I'm going to change it to log to the base, log to the of 4. Or my expression pattern should be, as long as it's 4 times as much, I'm going to consider it. Or 8 times as much in case when your differentiated genes are significantly higher. You say if it's 8 times, I'm going to accept it. I only want to get the really, really strongly upregulated. Now, on the other hand, if you want down regulation, and if the half down regulation doesn't work, you go with 0.25, which is a fourth, or a 0.125, which is an eighth. Okay, that's why you use a log 2, which means to say that this particular disease gene in the disease from a tumor, for example, is double or more than double, you say, okay, that is significant. Because if it's like, okay, this is a value 1 and that is like 1.5, is it really upregulated? Can it be buried in the statistical significance? And that's very important. Okay, and you could do all of this in a microarray, and of course this is a very small version of it. You could do, you could go into Affymetrix and say, give me a 10,000 a probe set of 10,000 oligos, and then I get a whole bunch of expression patterns. Okay, additional steps you have to do is normalization to make sure that the data is standard. And then once your data is standard across the board, then you can actually assume that the analysis has some statistical significance. And then what you can do is this is an analysis which is called as clumping, which is clustering, which is similar expression patterns have the same, and you're like, okay, these are important. And sometimes now, and this was not done during microarray, now when you have QIAGEN's, and several free pathway analysis software, you can take a bunch of genes, right? And that's important because what are they trying to say? They say we have a bunch of genes, and these genes are something that are so important that they belong to the same pathway. And then you say, oh, in this particular disease, for example, this pathway plays a major role, right? So it's a lot about understanding of something, and it all starts from here. And then you take 2 steps back, it all starts from being able to sequence a gene. Now, hopefully this will work. So StringDB is, I thought I'd throw it in here. It might seem a little bit out of place, but we'll do it anyway because I think this is a good StringDB. Okay, so this is a very useful a useful tool. So let's see, and I think stringdb.org. Okay, good. This one works, thankfully. Okay, so StringDB will actually give you a network of protein-protein interaction networks, and it also gives you, in terms of enrichment, it gives you things like functional analysis. So it's actually doing 12,535 organisms, 59.3 million proteins, and so this is a protein-protein interaction network. Okay. So let's take a look at this, and let's say protein name, let's see here, protein name, we'll call it, anybody's got a protein name you want to add into chat? Anybody's got an opinion that you want to share with us? Anybody working on a particular protein or cytochrome B? Very good, thank you very much, Michaela. All right, so let's put cytochrome, was it B or C? B. Okay, and organism, there's a whole bunch when you do the drop down menu, which you can't see here, but let's just worry about auto detect and we'll click on it, right? Let's go with Homo sapiens and we go select and we search. Now you'll see a lot of publications, right, with these things, and continue, a lot of publications, and actually, so now what you see is now let me make this bigger, and it's gone away, so let me bring it back. Too big. Okay, so here is this is not the entire pathway associated with this protein, but this is the nearest neighbors. Okay, so cytochrome B, I think that's the one. I think the red is the query, I think. Okay, so this is the red is the query, I think. Okay, and if I'm mistaken, I'll correct myself. Okay, so I think the red is the query. Now you see it is connected, like this is the first step connection, immediate circle. But here's the part that's really cool. See, you can kind of make it, you can just drag it with the mouse, make it big so you actually see what's going on. And so you can see that the red one is the primary query, and I'll explain what the point of every one of this is. And so you can see that they are in this first step, they're everything is very, very connected. Okay, now the question is, what do the connectivities mean? Okay, now all are not connected in the same way. Some are, like this one has a light green, a darker green. If you can't see it, don't worry for the time being. And then a pink. Then here you have this, and this is the original. This is the original. It's cytochrome B, and this other one. You'll see there's a black line, a light green, a dark green, a blue, a pink. So you've got all these connections. So what do they mean? So let's go down and see. It means that the colored is the node, that is our query. So that's our query, right? Then you see, if you have the white nodes, that means it's a second shell of interactions. This is, of course, nothing is white, so it's the first shell, right? It's the first shell. Then it says that if the nodes are empty, it means that protein, its structure had not been determined. Okay, and if the nodes are filled, that means that protein has either a structure that has been solved using X-ray crystallography, for example, or like cryo-electron microscopy, which is now becoming very popular for compounds that are not well behaved, or like a something that I do as part of my research, and that is derive or create using computational methods a structure. Now remember, I told you that what do these lines mean? The lines mean, for example, that these are connected. Where did that information come from? And you'd say that if it's a blue line, it came from a curated database. So this information, curated means a human has taken this database and put that information based on kind of a human knowledge. Okay, so okay, we found that interaction. So this is it's a very complicated, in the sense, a lot of effort goes into building something like this. So they've looked at the database and said, in a database, some protein interaction network database, they found it was. So that's the blue line. If a connection was determined experimentally, that's the pink line. If it was in the neighborhood, because they were in the same general area and you expect it to interact, it was green. If it's some sort of a gene fusion experiment, it's red. If the gene co-occurs, then it's blue. And then text mining. So even if you use artificial intelligence, machine learning methodologies to identify, let's say from the literature, some connection that you would otherwise not see, then it's light green. And then if they're co-expressed, it is black. And then if there is a strong protein homology, then it's just light blue or maybe purplish. Okay, so you can see that these are very well connected. Sometimes you might find a connection very tenuous. That means, what does this tell you? It means that it's been well studied, experimentally well determined, co-expressed, there even is a database of that information. So for example, you look at here between this, right, cytochrome B and this UQCRC2, and here you will get all the all the extra information. See that? It'll tell you all the extra information. These are the partners and they're color coded. So let's look at this thing which says UQCR cytochrome B C1 complex subunit risk, and it's mitochondrial. So you go here, and even this is mitochondrial. Oh, that's not the one. This is mitochondrial. This is the pink one here. That pink one here. Let's go to something closer. Let's say this is the one which is kind of yellowish, and that's I think is this one, UQCRC2. Does it? Yeah, UQCRC2, and that has been identified either by experimentally determined connection, and then you've got the blue, which means it is. I think it is. I think it's light blue, so maybe there's a protein homology between the two, or maybe it's in the gene neighborhood. I'm not sure of the color. And then the other one is light green. That we know it's also text mining. So somebody wrote a software which scoured the literature and identified that interaction. Okay, and so sometimes don't completely disregard the AI text mining component of it, for the simple reason that sometimes, curation means you got to do something by hand. A text mining means somebody's written a software which can do things maybe not as efficiently, prone to errors obviously, but there's probably something called as what is called a semantic relationship. Let's say you have two entities, right? Two entities, and the entities are approached. Okay, so for example, look at it in simple English, right? So I've got a pen here, right? And the pen is on the table. So I have one entity which is a noun, which is a pen, and then I'm knocking on the table. I keep the pen on the table. So what relates positionally, what relates the pen on the table is basically a preposition, right? English. Preposition. The pen is on the table, right? Um, so that's how these two terms are related. And so artificially you can also put in a whole bunch of keywords or key phrases that are semantic relationship, which basically relate to entities. And once you have those, you can write a program which takes nouns or gene names and things like that, and says, okay, the gene names occur in the same sentence. That doesn't necessarily mean they're related, but if these semantic relationships are present in the sentence between these two, you're like, okay, well, you can be pretty confident. Of course, a human would have to intervene and say for a fact, but you could be pretty confident that it was mined in that way. Okay, so that is that, and let me go back to the. So that's string DB. It's a very useful bit of information, and let's see here, PowerPoint. Now there is another type of study, and a lot of this, was actually represented in what's called a genome-wide association studies, and in the sense that which along across the entire genome, the upregulation, the downregulation, the presence of SNPs, the presence of single nucleotide polymorphisms, were all associated, and their associations could point to a particular phenotype. The easiest convenient thing would be the phenotype related to a disease, right? It's like that's what's important. So we'll see how that arose. And again, genome-wide association study, I think, personally, I think it has a lot of value, but it's kind of not being done very often. A lot of statistical genetics came through it because you wanted to look at these associations and statistically determine that they were valid. Okay, so all right. So in different ways, it's either called a common variant association study. The variant could be anything, a structural variant, a SNP, some difference between a gene, right? Or genome-wide association studies, GWAS, or whole genome association studies, WGAS, or common variation, CVAS. So, genomes vary between individuals in different ways. Okay, we understand that we vary because we come from different parents, so some things are the same, some things are different. Evolutionarily, we could have lost something, gained something, replaced something. So for example, even if you have a family of genes, you could go and do a study of the structural variants or the variants associated with that, and you'll find there are several thousand variants associated with particular genes. Okay, so for example, what kind of differences do you see? And single nucleotide polymorphism, a single nucleotide is different. This can result in a different protein, which gives rise to a different function, and that might be the cause of a disease. Now, this was a very a naive hope that you could identify a particular SNP and that was singularly responsible for a disease, right? The notion of genome-wide associations came study was because there's a SNP here, oh well, there's a SNP there. What is the association with that SNP? Are they both together? If one SNP was absent and one SNP was present, would it result in that in the disease phenotype? If they were both there together, if they were both not there, or that when I say not the gene, but that SNP, if that change was not there, would it be possible to do it? Would it not be possible to do it? Would it be associated with the disease? If there's one, are there three? Now what happens is the combinatorial aspects of the disease, those conditions become very difficult, and therefore they had genome-wise association. A lot of statistical genetics work went into this. Okay, there could also be a deletion. Okay, a single nucleotide could be deleted, and therefore that changes the entire translation profile into proteins. A large section of DNA could be missing from a chromosome. There could be insertions, replications, all of these. And they were looking for each of these to be with a disease. And then they said, well, are these variants now going to be associated? And do they combine while being associated to create a certain phenotype? And you can see, just conceptualizing this notion, that it becomes very complicated, right? Because now you can never really be sure. Here's something else. It's called the copy number variation. That means some genes in chromosome have more than the required number. That some genes are expressed a lot more, not just in terms of the levels of expression, but you have more genes of a particular kind that might result from insertions or deletions. Now, when I first started my faculty position at another university, I was with this group who I was supposed to be the informaticist for this group, and they were microarray experts. And one of the big papers that they published was copy number variations. Copy number variations in they were studying CNVs. So SNPs were supposed to be that one variation. CNVs were supposed to be the other variation, copy number variations, where it was like the number of genes were more and more. So now one of the interesting studies that came from this work was that in the United States we don't really have these, but in Europe, Poland, Sweden, etc., they have registries of different of children, and like almost like genetic profiles of these children are available. They collect them, store them, archive them for any kind of future work. And so sometimes, and then for research purposes, they de-anonymized the data. Like took away any name or any kind of thing that might identify location, address, and things like that, and they were providing it for research purposes. So this group, my boss then was from originally from Poland, but he was a faculty at Uppsala, but then he joined the United States university in the United States, and he was this microarray expert, and they were doing copy number variation. And one of the things they studied in terms of copy number variation was monozygotic twins, identical twins. And it turns out, based on this study, that they studied a bunch of twins, and they found out that even monozygotic twins had copy number variations in specific genes, and they used microarrays to test it out. Okay, and so it became a big paper. I didn't help in a big way. I just helped writing some software for them to do some kind of late stage analysis, and I kind of got on that paper. It's the eventually got published in the American Journal of Human Genetics, but it gained a lot of popularity to the point that my boss was interviewed by the New York Times because they said, oh well, monozygotic twins, identical twins are not quite identical. And I thought I mean, that was their selling point. That was the hook, where they say, okay, if they're not identical, then we have to ask ourselves, what is really an identical twin, right? And so this was, and they did it by showing from this registry of Swedish twins, monozygotic twins, of course, controlled for heterozygotic twins, or basically, or fraternal twins, or brothers and sisters, that twins actually carried monozygotic. But you understand this is that that's why you need the statistical analysis, because you have to compare it, right? Is this specific to monozygotic twins, fraternal twins, members of a family that are not twins but they're brothers and sisters? So that's why the statistical analysis has to happen. That just because something jumps out at you does not mean it's necessary. It's important, but it's part of the. But they actually showed that copy number variation uh, were present in monozygotic twins. Okay, so G was relatively new, but not new anymore, mostly defunct, because again, I think the point was made was that they were very that the associations were too complex to actually produce anything. So the method search for a SNP, right? Search for a SNP, and that, well, then you had what is called the kind of a SNP. You looked for frequencies, right? Frequencies. How many times did they occur with a particular disease? And then each study can look at hundreds or thousands of SNPs at the same time. They use this data to pinpoint the genes that may contribute to a person's risk of developing a disease, or somebody who had a disease, right? So this was like this supposed to be this early biomarker, and that is like, okay, this person has a SNP, and what happens, for example, if they have that SNP? Are they more susceptible to Alzheimer's disease, right? So that is the question, and the question is, can we do something about it? Can they be warned? Can they, have a lifestyle change that might help them a little bit? Yeah, this could be for anything, right? If for cancer, it is like, are there some behavioral epigenetic outside influences that might, force something, outside influences that might cause it. But then I think the point was that there was too much. The variations were too vast to basically just say, okay, I've got all of these, and all of these are in some way associated. As you can see, just when I gave you an example, if you had a three-letter sequence, AT, or four-letter sequence, ATC and G, in a combinatorial way, there's 24 ways that could be written. But if you went from to 5 nucleotides, then there's 120 ways, and you went to 6 nucleotides, it's 720 ways in which that can be written right? And so it became a combinatorial problem. And then its question is, are we assessing everything completely, right? Are you going to do the microarray of an entire complex organism? You can, you could, but the resources, statistical, etc. And then the question is, could you narrow it down to any specific disease? That became a problem. Okay, so GWAS is phenocentric. That means you identified based on an expressed characteristic. Two groups of individuals are selected. One possesses a specific trait, okay, a genetic trait, and another which does not. The latter is control group. And then the DNA of the individual is collected, purified, and then is hybridized on a SNP array, which is the same foundations of a microarray, where the SNPs are, where the regions, the oligos, contains a SNP. Okay, then 25 nucleotides long, and then these are for an allele. Allele is a gene that expresses the same the same particular characteristic, right? And then the remaining DNA, and again, you can go back to this discussion explanation. The remaining steps of DNA hybridization then correspond to those typical of a microarray, right? And now, based on the pattern you see on the microarray, you can then say, does this SNP exist, right? Or because of its complementarity. And the measure of the relative intensities were then sent to a statistical process, much like, again, the underlying methodology is still microarrays, but depending on what you put on there, the oligos with SNPs on them are, and you could put as many SNPs as you thought you want and see if there's an association with them. Okay, you learn some statistics like allele frequency, which is the population of an allele being studied. It is a measure of a variation at a position in a chromosome. Then you calculate the odds ratio. So that's the odds ratio in this case. Somebody with that allele or somebody with that SNP or somebody with that characteristic having a disease against somebody who does not. Now you know, right? Has a disease, has the SNP. Has the disease, does not have the disease, has a SNP. Has a SNP, does not have the disease, versus does not have the disease, does not have the SNP. Now I've given you 5 different ways, in which you can calculate the odd ratio, so you should be pretty familiar with it. And then you take the odd ratios, and then you would basically associate the gene variation with a disease. So for example, explains it a little better. You have the allele, you have that particular gene with that particular trait, and then you have the disease. So you say yes, have the allele but no disease, have the disease but no allele, and then no allele, no disease. You have these numbers, you calculate them, and in this particular case it's 0.42, and you can get the log of that. So it tells you it's literally a 42% chance of a real association happening. But now what happens is you have thousands of SNPs, millions of SNPs for a particular disease, and now all of them have to kind of get together in order to give you the probability that you're likely to have a disease. Other little definitions that you need to know: a haplotype is a group, so there's something called as a hap map, which was very important at some point, is a group of genes that are inherited together from a single parent, right? So they actually created like a haplotype, kind of like a genotype, which refers to the genetic. They created a haplotype. That's the definition that you would need. Then they created a international hap map. Now what happens is, as soon as they came up with a genome, that is a genome, they created, okay, let's create kind of a profile of that comes from one parent, right? Now it's decommissioned. And then it's like, well, what kind of variations can we see? And for those variations, you created a 1,000 genome project. And again, we have studied variations in 35,000 variations, but we were able to cluster the olfaction in various groups, like African Americans and blacks in one group, some Asian groups. In Europe, there was a lot of mixture. Okay, and so we used a 1,000 genome project, but they're saying, well, so the question about a 1,000 genome is that whoever was sampled when the genome first came out, was that a good enough representation? What happens if we took many? So when you took 2,600 uh, groups from, I think, 30 different families, I could have the number wrong. Then you really get a picture of the genome. How variant is the genome? Can we say that this particular set of associations, how many people are we really studying, right? Are we studying? And I'll give you one, and I think I'll end this class now because we've got to interview some personnel. But so I'll stop at about 3:30. But for example, in one of my studies, which is completely unrelated to genotype, we found something where we said that there is a part of a molecule, okay, that that can which is responsible potentially, okay, again this is science, we're probabilistic, is potentially responsible for somebody to smell chicory. Chicory is like a substitute for coffee. Very often the poor people in New Orleans and all, they use chicory instead of coffee, or a mixture of chicory and coffee, or it smells like maple syrup, or it smells like caramel. Okay, so the question is, why do three smells? Why are three smells associated with one particular part of a molecule? And the molecule is just a regular smell molecule, but three smells associated with that same, and I'm not talking about the whole molecule. I'm talking about narrowed down to a particular two atoms. They're not even bonded. But again, this is whole idea of molecular fingerprinting that I've been pursuing, published a couple of papers and gave a couple of talks to. So then you have to ask yourself this question, and that is, let's say somebody came from Asia, okay, and specifically from, if you came from a tropical area and you've never really smelled maple because it's not a temperate place, there are no maple trees, right? Or if somebody says, well, I recognize the smell of caramel, but I don't know what maple smells like, okay? And if somebody doesn't come from any other part, you might say, well, I know what coffee smells like, but I don't know what chicory smells like. Now, and this is not, had nothing to do with genetics, but it's possible. It's possible. Again, this is the theory that that kind of I'm conjecturing right now. No way to prove it other than kind of baby steps. Is that there's a molecule which has a particular smell, but three different smells have been identified with a kind of a signature and atom pair which are not bonded, but they point to that same particular atom pair. So then you have to ask yourself, in terms of smell, how many people can smell something? What happens, for example, if it smelled something, but you had no association with it? So if you came from a highly tropical place, even India is not highly tropical, it's subtropical, and you never smelled maple. Now there's maple in the upper reaches of India, but people have never smelled maple or maple syrup, for example, right? But I've smelled burnt sugar, so I know what caramelizing means. Or there's a very small group of people who would smell and know the smell of chicory. So now you've got to ask yourself, that means, is it possible that so much of our genetics and so much of our gene responses to olfaction and anything else is basically based on experiences, and how many of our structural variants in our genes, in our genome, like SNPs, etc., are very specific to a particular geographical location. Now, from a perception standpoint, from a psychophysical perception, we actually know that different people actually smell things differently, right? And that's just smell, right? Smell is not a disease. Smell is not anything, but something that has a lot more significance. And this is what is really crucial, because it's very likely that our human genome is not a true representation of the human condition. All right, let me see. I think one more thing that we can look at, one more slide, and before we quit. So again, here's the paper related to the 1,000 genome projects, but it said 1092, but the human genome, but this was when the paper first came out in Nature. But we've done some analysis and we found out that it really has about it was 2000, either 2600 or 2060 uh, different humans in different, very, very kind of cleverly identified communities that would give a very nice range. And I don't even know how but I've heard of the 100,000 genome project. Okay, so lots of genetic variations that we see, and again, this is a very difficult problem because this paper that we have worked on is purely based on the olfactory receptors on two chromosomes. We haven't done the entire human thing, but we see a whole bunch of variation, and it's a study worth pursuing. Okay, so the next part would be next generation sequencing, and this is actually a great place to it's a great place to stop. And again, if you have any questions, again remember this is a short week till Thursday, so if you have any questions, please ask them. Ask them. I think from whom I've heard, you guys are on the right track, and so let us again, the things are designed to make you think and learn things beyond what is taught in class, okay, which is what the master's or doctoral research process is all about, right? We are hooks and dracones, remember? Okay, I will see you next week, and again, I have to attend a couple of interviews that we are doing, so please indulge me, and we will continue next week. All right, all right. Have a good rest of your day. Have a good night, and obviously this impromptu email that I'm sending out with the class link seems to be working because they're not kicking us out. So I think that's that's another good sign. So I will see you guys. We are stopping 15 minutes early, so it's not. I'm not terribly delinquent in my faculty teaching responsibilities. All right, guys. Bye. Take care.